I always love those little shoes.
Oh, no, get back here. No, no, no.
Uh-oh. I'm
Well.
starting to have flashbacks.
You gonna close up the bin there?
Close it.
And then we'll secure section.
Raj.
Too much wrist rotate there, I suppose, for that guy. All right.
Okay.
Gonna drive backwards and I'll see if I can refine him.
Yeah. There he is. Got him.
All right.
Well, maybe we should open the tool tray.
No, because we have the coral in there.
You want to turn section on?
Yeah, go ahead. There we go.
Okay, Raj. I'll go push forward a little, yeah?
Yep.
And I'll just sit down right here. I'll try to do it gently.
Awesome. And just to confirm, you're full rack back still?
Yeah, I am.
Great.
I'm sitting down. I swear I'm going to sit down.
All good.
Okay. Opening. Tool tray out.
Roger.
Sonar looks okay.
Awesome, thank you. Take a look at the bubble. All right, you finicky little thing, let's put you in. Let's see here. How to do this instead. Oh, oops.
I'll tilt up for you.
Sure. Not really in the ideal position of this, so let's try this instead. All right.
Okay.
Once you get stable, I'll have you secure section there.
Oh, yeah. Okay.
There's an
Jesus. Just sit down.
I'm gonna go down on the delta bit. That's gonna give you better.
Okay. Ready to turn off the suction?
Stand by, let me give you more delta. All right.
Ready?
Yes. Let's do it.
Okay. Do you want me to slowly decrease it or just turn it off?
I want you to turn it off quick and then
Okay.
I'll try to jostle it in.
Okay, going now.
Okay.
0%.
Okay, maybe thrust up a bit.
What?
Oh,
I'm just gonna
Oh.
start.
close it then.
Get
Crushed it.
goodbye. Go to sleep, little urchin.
No.
That's disturbing.
So awful.
Nice job.
All right, and once you pull off, I'll put this section back.
That's sample 191.
191.
Why are we not getting you in here? All right, Chris, go ahead.
Bridge nav. 30 meters, 100.
Our scientist assurer, Tina, says, hail the pilot and co-pilot.
Yay!
That was fun.
It's always drama with the urchins.
Oh my, yeah.
That
Like
was skill.
What kind of urchin is that?
floaty. So I,
The crafty one.
Yep.
Versus the sinky kind.
I was going to say purple, so I'm glad we could all be helpful.
I'm looking it up.
How many feet do they have?
Many, thousands.
Thousands. Millipede, eat your heart out.
They all got little shoes.
I'm gonna look at the box real quick.
Sure.
So it's only the starboard one that creeps.
Yeah. Yeah, the piston, I guess, yeah.
Reg.
Starboard piston creeps.
It's in the Echinotheridae, Tramicosoma hispidum.
I saw that, yeah. Yeah.
Why do they have shoes?
They probably use those to grip the substrate a little.
And as status symbols.
Everyone needs to cover their socks with something.
What would those sea urchins be feeding on?
I think those ones are deposit feeders, so they're eating stuff out of the sediment. Although I think there was one that had something else in its gut, one of the ones we collected recently, but I don't remember exactly what it was.
Blacked it out.
Possibly.
I would have.
Here's a question in the chat. What are you listening for using passive sonar? Is it ship mounted or on the ROV?
We don't have any passive sonar.
We have active ones?
What? What? It wasn't for you, Bridge. Sorry. Yeah, I guess the closest thing we maybe have to passive sonar is if we use like an LBL system.
What?
Do we ever do that?
No.
No.
Long
I mean,
baseline?
yeah.
Long
I mean,
baseline, yeah.
Yes, I know it has happened, but not since, I don't think since I've been on the ship.
Yeah, it happened a long time ago. I
What's
think
the difference between LBL and USBL?
Well, one's a long baseline and one's an ultra short baseline.
And operationally, what difference does it make?
So long baseline are beacons that you place on the seafloor far apart from each other, and you kind of operate in the center, in between them, and you're able to triangulate your position.
You need at least 3 of them. You have to survey them into position.
Yep.
And you have to operate within those confines.
Whereas ultra short
turn into the slope.
baseline has a single head on the bottom of the ship, and it basically can, by measuring phase difference between its 3 or 4, 6 cents, however many it has, by measuring the phase difference, it's able to tell which direction the sound came from.
So another move in there, Chris,
yeah.
please.
Bridge nav, 30 meters, 100. So yeah, it's the USBL is able to locate the vehicles.
Sorry.
No, it's okay. I think I'll have you come back because every time we pull on the tether, it's making that pan and the tilt like go all wobbly.
Yeah.
So let's try not to yank on it too much.
It's going to do that anyway, though.
No, but every time we pull on the tether, like it gets tight, that starts to move more.
Yeah, but even with the ship heave.
Yeah, it'll also do it with the ship heave. So it's just like one less thing. Keep going, Chris.
Yep. So anyway, yeah. I think LBLs used to be much more precise. I don't know that they're...
I think they are very precise, but they just take more operational time.
Yeah, much more.
Yeah.
I'm not sure that they're that much more precise these days. Like functionally speaking, I don't know that it's really worth it in the end. I don't. Well, maybe if you're really deep, it might
Okay,
matter because
sure.
USBL falls off with depth.
Yeah.
But at the depths we've been operating at, I can't imagine a LBL being much better than what we're getting with our relatively new USBL.
Right.
But you can, sometimes they're popular for like ASV or AUV operations. Hang on, the ship isn't moving yet. Bridge nav, 30100. Yeah, they're sometimes popular for AUV operations because you can set them up and leave them, and the ship can go elsewhere and do other things. So.
You have a zoom on the chrysogorgia? Or
Video
whatever it
zoom?
is, maybe a tiny little metallogorgia.
Yeah, metallogorgia. Huh?
Yep.
Squat lobster. You have any more?
Great footage, thank you.
You have any more? I want the polyps. Give me the polyps. So those white things in the polyps, are those like for reproduction? Come on.
I don't really know. It would be hard to say without looking at them under a microscope in a lab. It could just be the way the polyp is showing up in the camera.
Okay, gotcha. If it was an umbellula and they were in the same, is that about the same place that they'd be, like sort of like behind the polyp?
Yeah, down in the polyp you can sometimes see egg structures.
Gotcha.
Yeah, when I took over for the dinner watch, there was an umbellula with a bunch of eggs in it.
Exactly, and
yeah.
I didn't really see the what they were saying, so I was wondering if it looked like what we just saw then.
Nah, that was like a clear, like little sac at the base of the tentacles.
Oh, gotcha. Raj.
Cool.
Cool.
It's really thick-looking sediment chute right here. Ripple marks coming down it.
We have arrived at the small wall.
Is that a loose rock
Small
on the bottom?
wall or big rock?
Not sampleable rock.
Oh, hey.
What about this rock?
Let's run into it.
Or is it too much of an overhang?
Yeah, maybe it's not the most comfortable spot for us to be in right now.
It's a nice looking rock though. Look at that.
Very angular, yeah.
I'm talking about the one right in front of us. It's angular. It's basalt.
Nice rock with a lot of chrysogorgia and a couple hemicorallium.
There's stuff on it.
I'm going to start coming up a bit.
Okay. Can you just push in on that rock? I'm not going to be able to give it any light, but again.
Yeah. I mean, if that's difficult, we can avoid it. It's pretty, well, it's pretty small.
It's tucked right underneath the pit of a cliff that we need to ascend now.
Okay.
Come wide. That fix of the zoom for a second there, I thought I had like the forward thrust on like really hard for a second.
Oh, right.
I was like,
Got a variety of corals. A hemicorallium, it looks like, on the right.
Yeah, hemicorallium. We've got a couple plaques, a couple possible bamboos.
What's that next
Nice
to the bamboo?
iridogorgia.
Yeah.
I think it was an anemone.
Yeah.
We're going to have to come up faster, actually.
You want me to back off a little bit,
100%.
Jess?
No.
Okay.
I mean, we won't feel it in time.
It was right at the top right here, yeah?
Yeah, let's see if it's the last ledge.
Is this the cliff? Or
No, this is
we're
just
far
a
away from
small
that.
cliff.
That cliff is 400 meters that we're going to go to eventually.
Yeah, that clears part of it. And there's going to be a steeper bit to the slope there.
Should we pitch any weights? Because that's the fastest I can come up.
No, we don't have to
Okay.
pitch.
No, I meant like for the future, we'll just go slow into it.
Yeah, we're just going to go slow into it.
Raj.
Here's the top of the massive flow. Fairly smooth. Finely sedimented, so you can't get coral on top of the flow. You can see the area of the current's really good. Coral like it. It's just there's nowhere to get a hold fast.
The shot in Atalanta looks cool.
Yeah.
Yeah.
Looks like a skull.
Or like a stingray body or something.
Oh yeah, I saw that too, like a mobile ray or something.
Highlight.
Tiny little coral.
Bridge nav 20100.
Rock face.
Meredith, are you still interested in water samples along the route?
I am. And actually, right at that cliff edge was kind of a nice spot for one. So we're probably still close enough. If we take one right here, we'd be just fine.
Sure.
Since we're right in that area.
Sure thing. Kylie, my favorite. Oh, go ahead. You full wide there on Herc there. Thank you. And Kylie, you full back.
Yeah.
Oh, that's not indexed.
Oh. One and two are popped, so any of the rest.
Sure. And look a little down there, please. Thank you.
Not yet.
Nothing?
Nope.
I don't think this one's rigged.
Yeah, it might not be.
All right. Stand by.
Raj.
Coming up on the winch. All right. So back row, you want to make a note that Niskin number 3 is not able to fire. All right, let's go for a 4.
You're good.
Okay.
That was sample 192.
192.
Thank you. Does that make Niskin 3 some kind of finger trapping booby trap when you work on the ROV later?
No.
No, it just probably means that the little bobbin was rigged in not the right way.
Either it's clipped into only one side of the monofilament, so it's hooked up on the other side, or some of them have like a metal loop in the back of them that you could accidentally clip the top into. Just push past that, the magnum. Thank you.
Here's a question in the chat about how big are the fish and sponges and any other things we've seen, and have we also seen any crabs? So I know we've seen some spider crabs. Yeah, you want to
Bridge
talk
nav
about the size
30100.
of the different species?
We've seen actually a wide variety of sizes. So a lot of the stuff we're seeing on these dives hasn't been huge. It's been kind of 10 to 20 centimeters, but on previous dives, we've seen some bamboo corals that have been a couple meters. Same with fish. We've seen everything from tiny little, kind of recently settled out eel-like fish to we saw probably a 2.5 meter shark yesterday. So there's a wide variety of sizes we're seeing.
And of course the lasers are spaced 10 centimeters apart, which is a little less than 4 inches. So that gives you a rough idea of how big the objects are that we're looking at. Or creatures, I guess. I shouldn't call them objects. Here we are in the kind of the caldera rim of the middle volcano, I think we're on.
So, oh, sorry.
I'm just saying, I'm not seeing any loose rocks, but I'm keeping an eye out.
Quick question for you, Beckro. As we go from waypoint 5 to waypoint 6, obviously we're going to slow once we hit the wall, or before. But as we go downhill from waypoint 5 to 6, do you want us to just kind of go fast downhill? You know what I mean? Like,
Yeah, I think you can do a jump, basically, if that's fast and...
furious?
Yeah, and then if it's all sediment, I say we do, you know, half a knot towards the wall.
Okay. All right, so we won't get as good of visual data going downslope, but if we go fast that way, just keeping that in mind.
Yeah.
It seems like the high priority is the wall, yeah?
Yeah.
Okay.
And bonus watch points if you can do a flip while going down the hill.
Well,
Olympic style.
only if we're made out of marbles.
There's not much on top except for snow and crinoids. Marine snow, that is, which is mostly calcium carbonate.
It's so sticky, I doubt you could ski or sled in it.
No.
I didn't know marine snow was sticky.
I was zooming the fish.
Ooze.
Well, I don't think it's sticky when it's, per se, when it's up in the water column, but when it's once it forms that mud on the bottom, it'll stick.
Raj.
We collected a sediment core during this dive, and we're gonna have to slice it up and like package it and stuff. So when we're doing that, you should come and
Oh, yeah.
touch the sediment. It feels like kinetic sand. No, did you ever play with that when you were young? Yeah,
Oh,
it's
yes.
very satisfying. So you should come hang out in the wet lab.
Okay. Video zoom.
Little chrysapathes there.
Okay, come wide.
Bridge nav 50100.
What are all the numbers that you say to the bridge?
Those are distance and bearing to
Oh,
where I
okay.
want him to go.
Cool.
Mm-hmm.
So like 50 meters and 100 degrees.
That's correct, yep.
Here's a question in the chat. What is a chrysapathes?
Chrysapathes is a species of octocoral. So the little one that they just zoomed in on is in that genus. They tend to have these little sparsely spaced bell-like polyps along their skeletons. And if you look closely, their main stalk tends to grow in a spiral pattern.
Is a lonely stalk crinoid?
Yeah, so I saw in the science chat that the
Metrocrinus?
yes, that one is a crinoid that doesn't have a stalk.
Yeah, that's a feather star, so it's one of the ones that can swim if need be.
Okay, cool.
So most of the ones you see just kind of flat on these rocks sometimes, those are the feather stars. The ones that have a defined stalk like this, these are the stalk crinoids. So this guy can't swim. But it can look gorgeous.
Beautiful.
Kylie, might have to get your bubble.
No, not at all.
This really evokes taking a walk in a snowstorm or just after a snowstorm.
No, I've got like off to grandmother's house we go, sort of like playing in my head.
Yeah, I'm thinking Robert Frost, but yeah.
Oh, yeah.
I like how quiet it is after a snowstorm.
Yeah.
It's so relaxing.
Nobody has their cars shoveled out yet. It's just...
I can always tell when it snowed at home when I first wake up in the morning because just it just sounds slightly different even from inside.
Really?
Yeah, like
For sure.
just the muffled, like the whole world has kind of a sound dampening blanket.
Yeah.
It's so nice. I was really missing it in Arizona, like all the snow, because I grew up in Iowa, so it snows like a bunch there every year. And then I went back in April, and there's a fresh layer of snow on the ground in April. And I was so happy to not live there anymore. I did not miss it anymore after that. I needed that trip to remind me. In April, that's so late.
I told myself if I do one more New England winter, I'm getting a snowblower, so help me, God. I'm a grown-up, I've paid my dues. I cannot suffer in this way anymore. I have an exceptionally long driveway, though.
Oh no.
I think that's fair.
Yeah.
Yeah, you deserve it then.
We always have the Puget Sound area catch-22 where it doesn't really snow frequently enough for there to be proper infrastructure for things like clearing sidewalks.
Right.
But at the same time, it will linger long enough just to make sure that it's obnoxious and dangerous when the sidewalks don't get cleared.
Mm-hmm. Mr. Professor Tim, why is it that small small mounds of snow melt faster than big mounds of snow?
Huh. And are you talking about like piles of snow shoveled into the Walmart parking lot? Or
Yeah.
Okay. I'm picking up what you're putting down. Okay. Yeah, it's probably just the self-cooling effect of a really massive surface area. I mean, sorry, lots of volume, low surface area. So, or a smaller surface area to volume ratio.
Because they just last forever, those big mounds.
No. And they find random stuff in them too.
Really?
Yeah, like all the trash that gets like, you know, shoveled into that pile by the big old snow plows. And
yeah.
you're like, oh, there's a random doll foot in here.
Yeah.
That's creepy.
They're never good.
Can we zoom around there just to see if there's any loose rock?
Yeah, I just need a little bit more time to get a little closer.
Take your time.
So my best find after a Massachusetts snowbank melt was a 1980 10-speed Fuji bicycle.
Ooh.
Dang.
That I reported to the police, and they were like, nobody's reported this missing.
Ooh.
So free bike,
Ownership acquired.
more or less in perfect cryogenically frozen condition. But it had clearly been part of that berm for a while.
Yeah. There was one that like melted in July, right? The last one in Boston. There was like a snow pile that stayed until July.
Jeez Louise.
Yeah. Or maybe it was late June, something crazy. Just kind of disturbing.
You think of the top or the bottom side? Oh, sorry. Didn't mean to scratch that. Because I think that maybe that one might be loose up here, otherwise poke down there, yeah?
Yeah, I think where you're at's a good spot.
So just drop down here?
Yeah, if you want to look down on the camera a wee bit. Any of these looking like what you want there, Kevin?
Yeah, anything that is potentially loose, like maybe these or maybe these, it's hard to tell.
Okay.
Yeah.
They might all be cemented, but fingers crossed.
I'm going to switch over to starboard bio box cam. I'll come down a wee bit too.
Wow, everything's bigger than I thought it was.
I just take your bubble there.
Raj.
Okay.
Let's
Do
see.
some poking?
Yeah, can you reach in that direction?
Yeah, I think I can probably reach this one. Maybe. Oh, maybe not.
I get kosher.
Let me see if I can. No, yeah, no.
Oh, wrong. can't see. Watch.
Between a rock and a hard place.
Say that again?
Between a rock and a hard place.
Oh, yeah, really. Okay, let's just see what we can do with this.
Bridge nav 20280.
I will try one more time with that guy. Do a little bit of a pull behavior.
Everything's okay, I'm just pulling up for fun. Because I think it's what you think is the right thing to do.
No, I think, yeah,
And everything's fine.
red judge.
Everything's fine, except I'm pretty sure that's going to be what you want soon. So I'm just doing it.
I think we're good, because
Yeah.
yeah.
What about that one? Yeah, right next to it.
Yeah,
I'm assuming it's also cemented, but
I think so. Let me try. Bleh.
done. And can we reach this one here or no?
Let's see.
'Cause that one on the forward cam looks pretty good. Both of these look a little looser
Okay.
on the still cam.
Go ahead and push on in a wee bit. Still cam must be lying.
Yep.
Looks like it's in the wall there, and I can't reach that one either. Maybe. Yeah, it's all up on all my joints there. Just out of reach. Go ahead and come wide.
I'm going to turn my heading
I don't think you got any room
to do it, yeah.
Well, we can, we got some time here. We can have Kylie kind of drive up a wee bit.
Yeah,
Check the other side of this.
take that one. Yeah, you can. Yeah, we can go down a little bit to the
Yeah.
other end of the hill.
Where's
Can we get a quick zoom on the white squiggle that's right under the arm?
Sure, yeah, zoom in.
Tube worm?
Yeah, it looks just like some kind of sediment-covered worm tube now. Thanks.
No problem. Okay. Coming up.
Sorry, I won't be able to look down on you here.
What was that? Oh, Rod, no worries.
Yeah.
Why? Why? Sorry, life is hard. Okay. Lance.
Bridge nav 20280.
So here's a question in the chat. Can you help with explaining the difference between bearing and heading?
I can't do it. Rod, coming up, coming up.
Yes.
Nice.
Heading is the direction the ship is pointing. Bearing is a direction relative to the ship.
I'm coming up still. I'm
Well,
gonna look at it,
yeah.
actually.
Doesn't bearing and heading differ also because like you could have like a crosswind, so your heading might be pointing like into the wind, but your bearing is like the direction you're actually going?
I don't
Yeah,
know
bearing
if I
is-
can get down in here.
Yeah, I guess that's maybe a more concise way to say it. Bearing is the direction that you're actually moving.
So,
Like,
Well,
sorry.
Okay.
hold on.
Sorry,
Yeah.
guys. Can we belay that talk for a minute, please?
Yep.
All right. So, Kevin, some of these spots are in a wee bit of a weird, tricky spot.
Right.
Maybe kind of like center screen on Atalanta to the right-facing wall there.
Yeah. Do you think does it look like there's anything loose on it? I'm trying to see.
I can't tell.
Yeah, it's kind of well cemented.
Oh, yeah. Maybe Kylie's coming up on a good spot here.
Yeah, right
The
above
backside
the
of the big
lasers
bump is.
now.
Okay.
Yeah, you're looking good, Kylie.
Okay, cool. I'm just going to be a little bit under you.
That's all good. I'll come up a bit.
One
That
of
one,
these look,
yeah.
look better.
Yeah. Good. Nice.
Cool. I didn't want to squeeze in there.
No.
I feel like Austin Powers, right? Like, yeah, ow.
Do you mind if I take your
No problem.
How about this guy?
Yeah, let's try them.
Gonna do one bump forward there.
Yeah.
Yeah. Oh yeah, I think we have plenty to play from here.
Fingers crossed.
Where's the bottom?
Do you want the right or the left?
Let's try the left one first. It's the better looking one if it works.
Sure. I think we can barely reach it. Oh yeah, we can reach it.
No.
Yep.
Oh, I think it might have moved a wee bit. Or maybe that was just, let's see. Kind of see it. Negative. Okay, fine.
Okay, let's
We'll try that one just to the right. Yeah.
I'm gonna pick up a little bit, Raj,
Sure.
just to put you back over.
It looks like that one should go, I swear.
It almost looked like you broke it in half from here.
Yeah, I'll watch the starboard there.
Raj.
Can I poke at it one more time? Do you guys mind?
Yeah, go for it. Show it who's boss.
Okay, let me just...
Actually, I'm gonna get my fingers cleaned. Okay, fine. Sorry. I really thought that one was going to come out after a third chance.
Is that, hold on. Is that piece off? No, that's all one big piece, isn't it?
Yeah.
Okay.
Yeah, I'll come down a wee bit again.
Okay. Want to try this one near the slurp?
Yeah, I think that was the other one that Kevin had.
Okay,
Yeah.
just...
All right, I think that's perfect there.
Just trying to touch something. Sorry.
You're good. It's kind of a precarious spot.
Yeah.
If anything, I can do it on the go.
Yeah, okay, good. Sounds good to me.
Gonna look a little up there.
Right.
Thank you. Great.
So Enjoy getting down.
No, that one's also in there.
Yeah.
Oh, I see what they're saying about being really twitchy.
Yeah.
Very interesting. What about the one up
4.
there to the top right?
Okay. I got to lift up. Nope. Nope. Okay, right. Okay.
Kevin, there's really no good spot here.
No, it does not seem like it. This whole,
I'm gonna lift up and sort of just look around.
Yep.
yeah, it was just,
Okay. Let's sort of.
Move up by that rock.
Which?
The one in Atlantis, Cam. Maybe there's some more broken stuff over there.
I'll take any direction on which one you want to poke, but maybe the one at the bottom here in front of these maybe? I don't know if they're too big or I just think it's...
well, can you zoom a little bit maybe?
Sure, yeah, go ahead and push in. And just hold there, and I'll just sort of like... I think that's all one.
Yeah, that seems like one big unit. That's just partly sediment.
What about the one with the left lasers on it? Well,
I don't think that's going to... Well, we can try to poke it, yeah.
Sure. Okay, come wide.
nice. Great. Yeah, okay.
Yeah, this is all, I think, a cohesive unit.
Roger. I'm gonna try poking this one over here, and
Sure.
then we'll probably just have to keep moving.
Yeah.
Unless, I'm sorry.
What about that one little nub and that little luge of sediment with the sediment on it, like that little to the left a little bit
That
more?
one?
Yeah, like that little piece. You think maybe that could be broken off?
I think that might be crust.
Just try it. Just poke it.
Oh, I can't reach.
I'm coming. Ooh. 16.
That's good. Yeah, I think that's all just...
You haven't even touched it yet.
I'm pretty sure this is just crust.
Oh, but we could try every other one.
I mean, it looks pretty hard on the wall there.
Okay, come wide.
Yeah, I think
Everybody,
we can keep moving.
let's
and
reset, yeah.
see if we find any.
Let's put everything away and just keep our eyes peeled. Okay, coming up.
Roger. Actually, Kylie, mind if we switch?
Yeah, sure.
We can do some poking.
You look like you were having fun there.
It was having fun. This is the best part.
Now it's like we're in a snow globe.
Yeah, that's pretty awesome looking.
Yeah, let's get out of here. Kevin, are we clear to start moving again, or do you want to try some more here?
Yeah, can we just look around this mound a little bit? Just get like a 360 maybe,
Sure.
or partial, not 360, but.
Yeah, sure thing.
Negative, Bridge. You're just looking for a friend tonight, aren't you, Oriole?
Interesting. Coming down a wee bit.
Did we poke in this spot already?
I'm not sure. We can retry.
Yeah.
I feel like we
Zoom on
have.
it. Yeah. Yeah, I'm trying to remember if that's the first spot we poked in.
Yeah. Oh, no, that was letting you get set up. Roger. All right.
Okay. Anywhere?
Yeah.
Raj.
Start poking around. I mean, this is what I first saw, but it looks a little more intact now that I can see it up close.
Roger. That's a big one.
Also, it's giant.
Yeah, that one's too big, I think.
Yeah, that one there. Little nubbin. I'll come down towards you.
Oh.
I don't know what that is, but it's,
We can look at it.
yeah.
It's like wood.
Yeah, a piece of debris or something.
Oh, I crumbled it.
Good. I had it coming.
I wonder if those are marks where the vehicle was sitting earlier.
Oh, maybe. Oh, yeah, we slid off the wall.
I don't know my own strength.
Okay, so, yeah, maybe we'll just start moving again. So, I can't find a single rock here that's not in situ.
Yeah. It's very deceptive, this area.
Yeah, the sediment makes everything look like it's loose, but it's not.
We can keep the arm out for a minute if you guys want.
Yeah.
What, the little piece? Rock. Raj. Okay.
Oh.
Guess we're going to have to order a pneumatic rock hammer for Herc.
Yeah.
That'd be kind of cool. All right, so Kevin, we giving up on the spot for a rock?
Yeah, I don't see any spots, so I think we can, yeah, put in the ship move maybe, and then while we're waiting on it, just poke around one
Okay.
last.
Do we want to? So we're at waypoint 5. Do we want to start moving to 6?
Yeah, I think we'll start the quick descent down to 6.
Yeah, so thinking this through a little bit more. I think we still have a little bit of uphill left on our sonars. So once we finish the uphill part, then I'll be comfortable with going 0.5 after that once we're going downhill.
Okay.
So maybe if we can start uphill at like 0.4.
Okay. over the hill, then warp factor 5. Raj.
Through the woods, yeah. All right.
Okay, new bearing 062.
Raj.
Roger.
065.
Gorgeous volcanic neck right here.
0.6.
What was it? What's
Let's get it.
65.
065, Raj. All right. You want to kill
0.6.
Alex to that guy?
Oh.
Thank you. 065, Raj. Go ahead.
Maybe right at the top here. Let's see.
You ready to move? Or are we playing with rocks some more?
I
A
think,
little bit of both.
I
Okay.
think we're gonna,
Like, yes, move.
Okay.
And
Gotcha.
if we see rocks to play with.
All right. Breach nav, 3 meters, 065.
Something biological has slimed the lens of the still cam, and now that's all it will focus on.
Oh. Ew, yeah, you're right.
First I thought it was a scratch, and then I saw it was moving. I was like, oh no, it's just been slimed.
It's just goo.
I'm glad it's just been slimed. Close to a stretch.
Yep. I don't know about that little nubbin.
I feel like we were, I don't know, it all looks very familiar. It all looks the same, doesn't it?
Yeah. I mean, do we want to poke around over here, assuming we haven't done that already?
Go ahead and stop the ship there, actually, real quick.
Huh? We're moving. Oh, stop it?
Yeah.
Okay. Breach nav, hold position.
We're gonna get Kevin his rock.
We're going to do it.
We're gonna get a rock.
If it takes us the rest of the watch. All right. Which one did you see there, Kevin?
I already lost. This way, maybe this one, or that one,
Okay.
or this one. Any of these nubs.
I think
Just
let's
poke them.
over this one here. Coming down a wee bit. That's about as
Give me the rock for Kevin.
I think the wee one will come.
Potentially. There's also this guy up here. He might be able to break off.
Oh my goodness, these
Yep.
rocks.
It's all attached. Ooh, I broke it.
Try one more time, Pate. If you're able to break it, then maybe we could break it off.
Is that what we want, though? Yeah. Give me it. No.
All right,
What
Kevin.
about this little piece right there? How about that? No.
Can you try to see that one move?
Yeah. Why don't
Oh,
you try
okay.
getting around here?
Yeah.
The rocks all told their friends about that saw.
Yeah.
They've all cemented themselves in place.
Switch over to boreal. All right. You're clear whenever you get this.
Okay, was something telestrated? Ooh, oh, come on.
Yeah, just if that will pop off.
Okay.
It looks like it has a nice handle at least.
It's
Yes.
moving!
Yes.
Wow! Good eye.
And all the pluses.
Okay. Is it okay for a zoom, Jess?
Yeah, go ahead.
Okay, go ahead, Kevin. Not Kevin, sorry, Tim.
I'm on it.
Words. Oh, I got a tiny, tiny little grip there. I'm not going to turn it around, I don't think.
We can
Am
just
I?
regrip it in the sediment there.
Yeah, we're okay. We're all right. I got
Yeah,
scared.
that looks great. Thank you.
Okay, come wide.
All right, I'm gonna come up a wee bit because
Raj.
might be able to clear it actually with the starboard bio bin. I don't know why auto dub is selected right now. Anything floating in starboard bio bin?
There's there's 2 corals, but they don't say they're floaty.
Roger.
B, D, and F are completely empty.
Okay. B, D, and F, Raj. I'm not coming out.
Whoa, Echo has a
Big boy.
has a boulder in it.
Okay.
All right. B, D, and F. Interesting that C is taken.
Yeah, it's going to be fun getting E out of that.
Not our problem.
Oh, man. That sounds like a geology grad student problem.
Sounds like Brandon issue.
It's okay.
Nailed it.
Wait, I looked down. Which one did that go into?
B.
Sweet. Wait.
Bravo.
Yeah.
Okay, cool. Thanks.
Yep.
And that's sample 193.
193.
I can't believe they just hammered that other rock in there.
Yeah. Can't you though?
That is taking up all the space.
It's not even seeding it properly, the seals.
So what do you think that rock sample was?
I believe it was a
Nice,
basalt
nice
from the
grab.
summit of
Sweet
this little
teamwork.
volcanic cone we climbed up. So a little post-erosional volcanic cone. By that I mean the seamount kind of eroded down to sea level and went below the water, and then
Valves
this
off.
volcanic cone came about.
Roger. All right, Chris.
Let's do it.
Yeah, we're ready to mosey.
Bridge nav, 30 meters, 065.
So there's some more questions about bearing versus heading. When a command is given to move the ship, for example, 100 meters at 180, does that mean the ship moves south 100 meters? If yes, how does that affect Argus, well, Atlanta and Hercules?
So yeah, that when I tell them to move south 180 degrees, or sorry, south 100 meters or 10 meters or whatever, the ship will start to move, and then we'll feel it. Then we'll see Argus make a similar, not exactly the same move, about 10 to 15 minutes later, depending on how deep the vehicles are.
Yeah. Yeah. There has been there was no Z bias when I came on, and it was like normal, like float, like sort of sinking a little. Actually, there was 2. The Z bias was 2. Yeah, we have everything, or not everything, but 1, 2, 3, 4. Yeah, all of it. Sure.
Back row, we're gonna drop a plate while we're here. Especially when we come up to a wall, we don't want to be sinky.
Yeah, that makes sense. Raj.
Right, I'll give you the graph valve here.
Thank y'all.
Purchase
You've been
on.
offering to the volcano for its kindness.
The principle of conservation of ballast.
Yes.
I mean, honestly, we have that giant rock over there, so We'll throw one, and then we might need to throw two. Because nothing's been thrown yet. Nice. Looks like we're going for two.
Do you want me to grab the other one?
No.
Okay, all right, because
No,
I
this is
didn't
great.
realize I was grabbing the bottom one.
Let's just pitch them both.
Okay.
I'm pretty floaty. Then anchor.
I know. I told you I was coming up as fast as I could before.
Yeah, I believe it.
I was like, what's a little slow?
Great. Thank you.
No problem.
Oh, yeah. Much better. Sorry.
Oh, yeah, you could. I guess, yeah, ROVGrab is an adequate thing for talking about that.
Are you talking to me or
No.
are you in the
No, I'm
background?
talking
Tim.
to Tim.
Raj.
We can only hear part of the half of the conversation.
Yeah.
Yeah, Raj.
Yeah. So anyway, yeah, so this is an example, too, of what we see when we're moving the ship around and how it affects the vehicles. So we've been sitting in position here for a while without any significant ship moves, and you can see how far Argus has ended up from the ship.
Gonna do a car wash.
I put that software up on channel 3 for
Yeah.
those following online.
Yeah, so the vehicles are like 70 meters away. So what I kind of try to do is keep track of where Argus sits relative to the ship, and anytime we get to stop, I can sort of recalibrate where it's going to settle, and then I'll make moves according to that, and sort of set it out. So if I want Argus to move north, I'll just kind of, or move to a target, I'll just kind of take a range and a bearing, figure out what that is, and then have the ship do that same move. And once we get close, I'll fine-tune it with smaller movements to the ship generally, if necessary.
But that gray line is the ship's track.
The gray line is the ship track. The blue line is the Argus track, and the turquoise line is Hercules.
So right now the ship looks like it's heading like due east, but it's
Yeah,
kind
so
of...
that's a good. So this is a good example here because our heading and our bearing aren't the same. There's no. 1 second. Speaking of, breach nav 30065. Yep.
Sorry. If you want to just throw a big one in, and then 0.4 it, and then 0.5.
Okay, you ready for that already?
Yeah.
All right. Breach nav, can we make that a 500 meter move at point at 0.4 065?
You get what you asked for, Jess.
Hey. Pedal to the metal, let's go.
Big move.
Where were we?
Big move.
I was very excited about the big move.
Our heading and our bearing are different.
0.4 knots.
It's like when you get like
Yeah.
maybe the craft.
So anyway, yeah, on here you can see that the ship's heading is about 90 degrees due east, but we're heading up here at about this direction, 065. So I put in, well, a 500 meter move now, so it's going to go out forever. Yeah, so you can see that the heading and the bearing don't necessarily have to line up. Generally they will if we're cruising, like doing mapping or transiting or something, they'll be more or less the same. But when we're doing this, it's important to keep the ship's heading into the wind and into the current to reduce the amount of force it requires to hold station. So for an entire dive, our heading might only change a couple degrees, but
Yeah.
There's one more question in the chat related to heading and bearing. Do the laser dots have anything to do with that? That we see, the 2 green laser dots?
Nope.
No.
The 2 green lasers are mostly for the biologists and geologists in the back row. That's so we can size what we're working with on the seafloor. So those are exactly 10 centimeters apart, and that gives us a rough estimate of how big anything we're picking up is.
But they, oh, sorry. The dots do stick straight out of Hercules, so I guess they could be like the heading of Hercules.
Here's a science-related question. Have you ever seen something get eaten on camera?
Yeah, predation event.
Yeah.
Biological euphemisms.
Sea stars eating polyps
Yeah.
in perpetuity.
Yeah, I've seen a lot of like the sea stars eating polyps. I've also seen a very dramatic lingcod eats a
We saw the
halibut
ground
nearly
polyp.
its own size.
I saw an octopus eat a crab once.
Ooh.
Whoa.
Interesting dynamic, power dynamic. Were you diving when it happened, or was it like here?
No. I was scuba diving. It wasn't.
Did you feel the sense to intervene?
No. I think the octopus was kind of showing off for me. It's like, watch this, buddy.
This could be you.
Exactly, you're next.
So just passing through, really. Can't stay for dinner. Sorry.
Yeah, the octopuses of the Northwest are.
Bridge nav, increase speed, 0.5 knots.
I had one.
Warp factor 5.
Sorry, Tim.
Oh, I've had octopuses like remove pieces of equipment.
From you?
Not
Whoa.
from. Well.
Fasten your seatbelts.
Fasten your seatbelts. 88 miles an hour, crank it up to 1.2 gigawatts. Here we go.
Yeah. We have a new George in training up there.
Trolling.
Yeah, I know. Got to be careful. .8 miles an hour. This baby will take off. Or 0.88? Yeah, that's probably closer.
What did the octopus take off for equipment?
It was we call them clod cars, but they're basically like pieces of gypsum or clay that dissolve based on how much current speed is going. So you basically weigh the chunk of clay, put it out, and then for a certain amount of time, and then bring it back in and weigh it again. And we had dozens of these things out, and I put one near what I thought was kind of an octopus's den, or not knowing it was an octopus's den. And then the next day, I mean, this is like, we've got like a steel pin in the ground with like cable tied to this thing to a brick, and it's like cable tied to the steel pin, and the octopus had somehow like undone the cable tie and like moved the thing
What?
20 yards away.
Get this off my lawn.
Exactly, right. That was weird. I had another one start grabbing an underwater camera in a housing with a framer on it, So a big sort of steel arm that frames up the photo.
Yeah.
Like, I set it down and I was doing some work and I looked around and the octopus was like dragging the thing off. So that was like, that one was kind of shiny. And I had a giant Pacific octopus actually grab me one time.
Really?
Like from behind.
Ooh.
Yeah.
Was it scary?
I screamed. I mean, I didn't quite understand what was happening. It's sort of weird, but I was moving along a transect, a line along the bottom. I was taking photographs at distances based on the measuring tape that we'd laid out on the bottom.
Mm-hmm.
And I went to move from one place to the next, and I kind of like moved a little bit, and then kind of like slowly slid backwards, and was like, "What? That's weird." And that can happen. You get your fin caught on some seaweed or something, and some kelp. And so I tried it again, and I moved forward, and then I kind of slid slowly back to where I was.
Wow.
And then, yeah, and I couldn't, and so then I tried to do a few things, thinking that one of my foot was caught on something, because it happens. I sort of kind of wriggle my way out of it. And finally, I kind of pitched my head up and then looked down the length of my body, so like chin down, and I'm looking down at my feet, and I see an octopus with 4 legs on the ground and 4 legs wrapped around my ankles.
Oh my God.
Wow.
I know, right? And at that point, I screamed and like jerked and twisted.
Yeah.
And then the thing let go of me and just sort of sat there.
It's like a deep sea toddler.
Yeah, it really was. It was, yeah.
Like, it's almost like the start of a horror movie, right? They're like pulling you under the bed.
Exactly right.
You know.
Dragging me back to its lair, right?
Yeah. My octopus teacher no more.
No, and this was, the size of I feel like the thing would have easily occupied the size of a card table, like top, if it sat.
Wow.
Yeah, it was a big, yeah.
Big Larry.
Yeah, probably a female. I didn't get a look at the one arm that will tell you that, but yeah, she had her own den with some eggs in it. It turns out, like at the other end of our transect, and we were definitely in her neighborhood.
Dang.
Wow.
That's scary, but great story. And glad you're here to tell it.
Oh yeah, I wasn't necessarily worried after that point. In fact, the second half of that story is that my good friend and longtime dive partner Kevin Turner, who was getting his PhD at the time, and he was farther down that transect line, and I thought this was going to be great because the octopus starts moving its way down the transect line towards Kevin, and I'm like, my mask is flooding because I'm laughing and smiling so much, like this is going to be hilarious. And my because what Kevin was doing was like looking at underneath the tape. He had to lift up the tape and write down every 10 centimeters what was underneath the line, and so his face is like right up next to the ground. I'm like, this octopus is going to be all over him, and I'm going to get it on film because now I have the camera out and I got it on video. Unfortunately, Kevin like saw it when it was like about a foot away. I know, and then it pushed back and kind of freaked him out, but it didn't grab him, which, what are friends for, right?
Right.
What color was the octopus?
Well, we were about 50 feet down, so pretty much everything's gray.
Oh, okay.
In the Salish Sea. But yeah, they tend to be kind of a bright red or pink. The giant Pacific octopuses do.
That's cool.
I don't think it was trying to eat me. I kind of think that it saw me sort of from behind and cross-section and didn't think I was as big as I was.
Might have just been thinking, why does this black rock keep moving?
All right.
Have we used Norbit at all on this dive?
Yeah, it's been running for the entire dive.
Oh. Can we
Chris
see
is
it?
just looking at right now.
Can we see it? Chris, what computer is it on?
Ross Proc.
Okay. Give me a few minutes. I think I can make that happen.
It's an oddly flat feature, this. Huh. I didn't do a good enough car wash. Chris, would you mind bringing up the map again there, please? So, yeah, I guess for the straightaway, we could, once the ship gets to the contours that start to step up, then let's kind of let it swing out a bit and then slow and all that jazz, you know?
Okay.
Thanks. Yeah, just looking at all this layback right now, I'm like, oh.
Yeah, I'll put what I'm going to do, how about I put a ship move in that is the appropriate length? Well, this one's 100 meters short, so that'll be a good time to reassess.
Yeah, that sounds good. Even before we get to like the steep stuff, but along this part here, just let the layback settle out every couple 100 meters.
Okay.
Thanks. I guess we're gonna get.
Or maybe, yeah, we could even, maybe the last 100 meters or something too, we'll slow down to 0.3, let it start to come out, and then we'll stop. And
Sounds good.
Norbit is now up on channel 3.
Raj.
Norbit.
Norbit cowboy.
That's right. Yeehaw.
Giddy up.
Whew, another steep, look at that thing.
Yeah. I find this contour map has been a bit deceiving because it's like we go up more than 10 meters. I know it's the course map, but
The map of lies.
exactly.
You got a slurp jar, slurp hose in your,
Yeah, it's okay. You can leave it.
Oh, okay, I'll leave it.
Yeah, I just wanted to look down more without. Sorry, just gonna see blue water otherwise.
Holler porch jewelry.
Yeah, we want to look at all the bedazzled things.
This is where that pan and tilt would be nice. 45-degree angle out in orbit. See what's coming?
Oh, nice.
What's the scanning sonar called?
Which one?
Sorry, the one on the screen right above Chris.
It's the mezzo.
The mezzo.
That's the sonar computer.
Oh, so mezzo.
Yeah,
Right.
that's the mezzo, and this one's the Sea King Tritech on engineering computer. Yeah. Wow, what's that?
That's the front porch.
Ooh,
Got a little carried away with the zoom.
it's like being in the future. Really. Pixels for days.
Yeah.
It's pretty.
Do we have any questions in the chat?
Yes, I am getting to a bunch of questions. So here's one, actually, about the collection boxes. So why are the side boxes labeled A to F, but the front ones are Omega and other, and not G and H?
Because small joys make a big difference at sea. And creativity is not lost on ROV pilots. And because we can, mostly. There's no logic to it as long as we have a distinct label. It doesn't matter what it is. They could have been numbered. They could have been symbols. They are symbols. They could have been, what else could they have been? Tally marks?
Colors?
Yeah, could have been anything.
Honestly, we could have put emojis up there. It really just depends on however we label it in the
Yeah.
data logger, for the data loggers.
As long as they know what goes in which bin and we give them something to reference what bin it is. We could have put stickers in them. A different season sticker for every box. Huh?
That's a little hard. But I agree. We could do a bunch of things.
It'd be weird if the naming I don't know, was changed on starboard, like between letters and something else, right? That would be strange, but the starboard bio box and the forward bio box are like distinct places, so.
Did that go in box 1 or A?
Yes.
Yes, which of course the answer is yes.
Yes.
Or box A versus box alpha.
Right.
Yeah.
It's all in good fun.
Cool. So there's another question in the chat. If there is life on the moon Europa, what does the team think the most likely class of animal they would see if Nautilus was launched under the ice?
Something that's not on Earth, my assumption.
Astrobiology.
Unless you got some astronaut jellyfish, which would be awesome. So I don't know. It'll probably be some kind of amorphous mass of cells.
Chanel's the one on a NASA project. So what do you think?
Yeah, I actually work on a team at NASA where we're designing a potentially life detection instrument designed specifically for Europa. And if there is life there, we think it's very small, probably a cell, that kind of size. We had to read through the Europa lander report, which is a report that a team of NASA scientists make with basically the science goals for what a Europa lander would look like. And it would have an instrument on there that could detect cells around like 0.2 microns. So they're expecting something really teeny tiny. Yeah, probably something similar to what life looked like on really early Earth. So it's thought that perhaps underneath Europa's ice shell, at the bottom of the oceans, there's hydrothermal vents. And hydrothermal vents are thought to maybe be where life started on Earth. It creates this awesome chemistry organic soup that is a great recipe for life to come about. And that kind of life is they're tiny little cells. So yeah, that's what NASA is designing their missions for, but no one really knows. It's a long-winded answer to say it will be small.
Oh, super cool that people are thinking about this.
Yeah.
And how to sample it.
Yeah, oh my gosh. And there's thought to be like a ton of sulfur there too, which like hugely messes up a GC graph. So there's like how to remove sulfur, and the radiation on the surface is also pretty gnarly because of Jupiter. So there's all kinds of things that we've been having to think about that are super unique to that environment. And it's like the ice is just, there are these things called tiger stripes on it, and it's like these openings in the ice, and they're just super gnarly and collect a ton of sulfur. And basically it's super hard to send an instrument to another planet. So NASA needs a lot of scientists. So all you people listening, should check that out because we need a lot of help.
One of the big engineering challenges for building instruments to do that kind of work is how do you prevent human cross-contamination with microbes from Earth so that you know what you're detecting in the exoplanet or on Europa is actually from there and not from just a hitchhiker.
So Chanel, do you think your current work on the Nautilus or working with and understanding ROVs can help NASA's future dive missions to Europa?
Oh, absolutely. I mean, it's kind of the same thing, right? We're diving into an environment that we don't know anything about. No one's seen the seafloor, right? And it's going to be the same thing when we go to Europa. So a lot of the operations that we do here on Nautilus, NASA is taking note of. And actually, NASA projects have used the ship to run simulation missions for like a mission to Europa before. So actually it's very relevant, and NASA has used Nautilus for that exact purpose. So tons of parallels that you can draw with deep sea exploration and space exploration.
So Chanel, how did you get involved with NASA? How did you get to learn about what you're learning about, and how did you become interested in Europa?
Yeah, that's a great question. So when I was in high school, I was the photo editor for my newspaper, high school newspaper, and there is a NASA field team that was going out to craters of the moon for 2 weeks to do basically what we're doing here, except in a surface volcanic field. And they wanted a high school student to come along and be a part of a video series that they were making for like K through 12 education. So I got invited as like in a journalist aspect to just be like a 15-year-old who doesn't know anything about that kind of science and ask questions. And then I ended up getting like super involved in the data collection, and I liked smashing the rocks and like taking measurements of all of the basalt. And then they invited me back for some reason and added me to another project. So I just kept working on it all throughout the rest of high school. And then, yeah, the rest is history. But there are a lot of opportunities that you can get involved with. NASA in high school, they actually have a high school internship program, which is pretty cool. There's a lot of like remote opportunities that you can pursue. And if you're an undergrad, there's so many opportunities you can pursue to get involved in NASA. So they have like robotics challenges. So if at your school you have a robotics club or like an engineering clubs, you could see if you can start one of those like NASA challenges that they send out. There's also internships that you can do. And honestly, just cold emailing people at NASA is something that I've done before and I do pretty often. So if you're looking through papers and you find a topic that's interesting, I just cold email them and either ask for the paper or ask for more information on it. So it's accessible to everybody.
So there were a couple questions about how deep are we right now, and we're about 1,635 meters. And there was another question about how deep can Hercules go, and Hercules can actually go to about 4,000 meters, but our other ROV, Argus, can go down to about 6,000 meters.
Yep, that's correct. We're limited by the depth rating of our instruments and our foam pack.
This almost looks like the surface of the moon.
Yep.
Yeah.
Not that I've been there, but, you know, from what I've pieced together from TV and movies and the backs of baseball cards.
So there's a bit of pretty consistent flat surface, same kind of structure on both sides of these volcanic cones. It's just these little pitted holes with bioturbation. Rare sea pen. Rare sponge. So we're currently cruising along pretty quickly.
Yep.
Trying to get to a more dramatic cliff face, which we're hoping will have some cool critters on it.
We're hoping to see it on our watch, so
Yeah.
that, you know, we're not the taxi driving watch here.
Here's another question in the chat. Do any of the live samples survive the journey back to the surface? And if they do, are you able to keep them to study them?
Quite a few of them do. It depends a little on how cold we can keep those bio boxes. So as long as they stay nicely sealed, we will get them back alive. There hasn't been great success at keeping, well, until very recently, a lot of these deep sea creatures alive long term in shallow water, although that is something that's an active area of research for a variety of reasons. And they're getting better at it. So for instance, the Monterey Bay Aquarium now has a deep sea exhibit that has a lot of deep sea critters in it that are alive and well. But that's not the focus of this expedition, and we don't have any of the equipment required on board to undertake that. So.
So what is the purpose of this expedition? For example, why did you decide to grab that rock, Kevin?
The purpose is threefold, twofold, manifold. Geologically, we're trying to understand the age and origin of these volcanic features. So the Johnston Atoll unit of the Pacific Remote Island Marine National Monument is loaded with a variety of different volcanic features like seamounts, geos, ridges, etc. So we're trying to uncover how old they are and how they formed, and we do that mostly through the chemistry of the rocks. On the bio side, we're always looking for deep sea exploration to try to characterize what kind of diversity of species we're protecting in these areas and make sure they continue to be protected. And then Meredith can talk about the eDNA goals.
Yeah, we have a couple of biological goals. So myself and Steve Oskovich are undertaking a study to better understand the biodiversity on these seamounts, but from across the whole Pacific, and what we would call biogeographic breaks, which are differences between different seamount communities, what those are and what causes them. And to do that, we're collecting the corals and sponges so that we can get them correctly ID'd to species and also add them to a genetic database. And then that genetic database is what we use as a reference for all of the environmental DNA samples we're collecting. And the nice thing with the environmental DNA is that it allows us to get a snapshot of overall biodiversity of everything that's within an area, but without having to go and sample every single animal. And so we're using that as a technique to look at different seamount communities from across the Pacific.
So Meredith, if you took a Niskin sample out here where there's not really any corals, but there seems to be a lot of, I guess, in-faunal animals living in the sediment there, would you pick that up? Would you be able to see, I don't know, worms and?
A little bit. We actually don't pick up too much in fauna because there is a nice break between what you're getting up in the water column and what you're getting right at the sediment. We would pick up some of them as long as we run the right markers. So the markers that I'm typically using are just for fish and for octocorals, but I do have primers that would pick up sediment in fauna if we were to run them. And actually out here we'd probably pick up stuff because I've seen, with the coral primers, I've seen 4 umbellulus so far, so there are some sea pens out here. It's actually really fun to watch. I've been partnering with Nautilus since 2016 to collect eDNA, and often we'll take what we think is going to be a blank or a control sample out in sediment like this, and it's surprising just how many times that sample turns out to be full of sea pens.
Yeah, it's a really powerful technique, particularly if you never see a sea pen while you're driving around. You know that, I don't know, maybe they're withdrawing into their protective tubes or something. Or you just missed them because they're not very dense. But they're there.
Yeah, and some of them can bury themselves in the sediment, so.
Can eDNA detect gametes, so sperm and egg in the water, eggs in the water, and identify them as gametes?
You could detect them. You would have trouble identifying them with gametes unless you're using some particular primers and techniques that would allow you to separate the haploid genomes that you have in gametes from anything else. But we usually use mitochondrial-based markers, and so you really wouldn't be able to tell. So you'd pick them up because they'd get onto the filter, but you wouldn't be able to tell that they were gametes per se.
Gotcha. Yeah, I was just thinking that might be a useful way to detect something about the life cycles of some of these animals. But if it wouldn't work for that, then it wouldn't work.
There are ways you could do it, but you'd have to do it in a very targeted way. So if you knew things about the structure of the genome that would allow you to detect, say, sex-based markers, you could design qPCR assays, but that would be a very, very targeted study rather than a broad study.
Which means you might get a whole lot of nothing for a long period of time.
Yes.
Bridge nav, 105 meters, 065.3 knots.
Sorry, do you actually mind if we let this layback settle out before we get to...
Okay. That'll put us right about here.
Mm-hmm.
We can stop sooner if you like, and I also slowed down.
Okay, yeah, if we stop a little sooner, then yeah, then we can keep on that move then.
Okay.
Okay. Yeah, this will take like 10 minutes to settle.
Yeah.
Sounds good. Thanks, Chris.
There are a couple questions in the chat about what have we seen on our dive. So have we seen any fish this far down? Have we seen any whales? I know we've seen a tripod fish and some eel-like fish called halosaurs, right?
Mm-hmm. Yeah, we've seen a couple different eel-like fish. We've seen some cutthroat eels and halosaurs. And we have been seeing a lot of tripod fish this dive. They've been really common. Yesterday we saw some sharks. Might get some more today, hopefully. It'd be fun. So yeah, a wide variety of fish at this depth.
Yeah, we haven't seen any whales. And I believe we said whales don't really dive down to this depth.
No, not to these depths. The beaked whales can dive quite deep, but I think we're deeper than they go. Oh, I stand corrected. Beaked whales could go up to about 2,000 meters.
Wow.
So we could technically see one.
Can we just put a GoPro on them and then it's our exploration?
Yep.
Critter cam.
Yeah. Really deep diving critter
Yeah.
cam.
And that's the skulls that we're seeing on a couple dives, several dives ago. It was actually a beaked whale skull. I believe there might be an image of it coming into the highlights if it's not already up there.
What's the rate of deposition for the manganese crust?
In this area, somewhere between 1 to 5 millimeters per million years.
Wow. Wow.
1 to 5, how, what unit is that?
Millimeter per million years.
Ooh, mama.
So,
Real slow.
yeah, wow.
So that skull had at least a millimeter.
Yeah, so also apparently there's some more work done that microbiomes can actually facilitate ferromanganese deposition by changing the oxygen and pH levels right around, a surface. So it could also have been sped up in this instance because it was organic matter originally.
Wow.
Or it could be a million years old.
Very cool.
That's incredible.
When I write my science fiction novel about how whales eventually recolonize the surface of the Earth, the terrestrial, this is where the DNA comes from. It's from inside of a ferromanganese encrusted whale bone from 2 million years ago.
Oh, really?
Look at this gorgeous shrimp.
I don't know, I'm just putting that together in my head right now.
Oh, Reg, go ahead and push on in there, please.
Come on with Chanel.
That's a good 15 centimeter long shrimp.
We get her, or is she?
It's not
Maybe,
using...
maybe we need a tighter zoom on the
no,
fluffy
Yeah,
feet
go ahead
or
and push
what?
on in a bit more there.
Yeah,
There we got
it.
kind of gallops almost.
There.
Yeah, that's the thing. It swims with his back feet and gallops with his front feet.
All right, full wide, please.
Got him.
Don't ever take invertebrate zoology.
No sponge.
Yeah.
Lowly sponge.
The fiber optic sponge, hey?
Seemed like it. Couldn't see the base, but the upper structure looked the same.
I believe you.
If I could bet a nickel.
But only a nickel.
Only a nickel.
You think eventually they'll be like, if I could Venmo you a nickel,
Not crypto.
I could Cash App you a nickel.
There you go.
Here's a message in the chat. Can eDNA be used to extrapolate population numbers?
Sorry.
That's actually an active area of research right now. So the Northwest Fisheries Science Center, where I work, has a study ongoing in hake, where we are using eDNA to try it paired with acoustic signals and some fancy modeling to try and get eDNA so it can be somewhat quantitative to how many hake are in an area. That's taken a lot more work than, say, what I'm doing. So what we're doing here, we're not able to actually quantify how many coral are in an area. There's just too much biological variability. But there are active research projects right now that are trying to, for individual species, be able to quantitate relatively how much biomass there is of that animal in an area. Going back to whales, I've been doing a little digging back here about the Cuvier's beaked whale, and apparently very recently one set a dive record where it was down for almost 3.5 hours.
Wow.
What's normal for them?
Those animals will actually pretty regularly dive for about 90 minutes down to close to 1,000 meters.
Oh, wow.
They apparently have muscle tissue that's just been really evolutionarily developed to harbor as much oxygen as possible.
Mm.
They have myoglobin as their oxygen carrier. I know that's true. I think elephant seals have that. They've got a different, you know.
I'm not sure. The whole article there was behind a paywall, so.
What's the
The
difference?
publishing industry strikes again.
What's the difference between myoglobin and a hemoglobin?
So heme is an iron-bearing protein that binds oxygen, and it's sort of like the major component of red blood cells. Stop me if I'm wrong.
No, I think you're right. It's the Uber of red blood cells.
And I forget, it's like one heme protein can bind for O2 molecules or something like that. But myoglobin, I think, can hold on to more. And I forget what myo means.
Mayonnaise.
So hemoglobin is a tetramer, so it's got 4 ring structures that each have an iron, and it can bind 4 molecules. Myoglobin has a single heme group, but myoglobin tends to be located mostly in the muscle tissue, whereas hemoglobin is in the red blood cells and circulating.
Oh. Rog.
And myoglobin has a higher affinity for oxygen, so it binds to it tighter.
Oh. I'm glad you followed that up. I was going to say, what do you mean? But it just binds tighter.
That means it's less efficient at releasing the oxygen in certain parts of the body because you have to have more energy to release. Is that
Yes.
why hemoglobin would be preferred?
Yeah, and hemoglobin actually, the way it functions, the more oxygen has been released, the more likely it is to release the other oxygens, and that's one of the things that gradient allows it to be an efficient transporter around tissues. And so, yeah, it reacts totally differently.
Cool.
It's related to pH too, right?
Yes.
The shape of the protein changes with changes in pH, and it dumps oxygen if the pH is low. If memory serves, again, I'm trying to remember.
I don't know. It's all
It's the Bohr effect.
The Bohr.
B-O-H-R.
like Niels Bohr?
Christian Bohr.
Oh, Rog.
I was gonna say it's got to be another Bohr.
Yeah.
Yeah.
I did a program on Niels Bohr, a project on Niels Bohr once.
Yeah, the
Rainbows.
shift in oxygen dissociation caused by changes in concentration of carbon dioxide or the pH of the environment is the Bohr effect.
Jess, if you weren't applying your interest and background in this, what do you think you'd be doing? Because you've got a strong biology background and obviously a strong engineering skill set. Did you ever think about going into medical type more stuff?
Yeah, actually I really like a lot of the medical technology and industry. One area I really am still fascinated by is bioprinting. Right now it's like still a relatively nascent field, but yeah, like 3D printing for organ scaffolds and things like this. I think that is also very fascinating to me. So maybe once my knees give out, then I'll go into that. You know, as you're on the ship for so long, your knees will start to go.
Let's be honest, it's going to be your wrist first.
Oh, shots fired.
Too
Probably.
soon.
Too soon, my wrist is still not healed. You know, so I'll 3D print some tendons for myself. But, you know, it's like, it's such an interesting question, or an interesting problem, because if you think about organ replacements, that, you know, if you could 3D print a lot of these viable tissues, you could save a lot of, have a good impact on the world. But then also you have all these questions of, you know, providing, if you have just one solid homogeneous tissue, then like, how do you put in vasculature and stuff like that as well? I used to not be as interested in the human aspect of a lot of biology, but more and more recently with all this 3D printing, this seems like such an awesome future.
Mm.
But I guess a lot of the human medical technology, for lack of a better word, there's a lot of really interesting innovation with, like, you know, it's like a form of exploration in such a really confined space.
Mm-hmm.
The process to get a device implemented, you know, takes a decade, 7 years or so, which is for good measure. So there's this catch-22 of, you want to see your device implemented and utilized, and doing it somewhere like in the ocean is really easy and quick. But I wonder how that process of waiting and, you know, constantly innovating, but for half a decade at least, what that feels like, you know.
And endoscopy is when you put a camera somewhere, right, to look at it.
Yeah. Yeah.
My dad had a heart attack recently, and they put in a bunch of stents through his veins. They didn't have to open him up or anything, really. And I'm wondering, like, I didn't know that was like technology. Last time I knew somebody that had a heart attack, it was a much more invasive procedure. Mm-hmm. And I bring that up to say that I realized then, like, oh, there's like all different types of technology that could be applied to different industries if there was somebody with both sets of knowledge, right? Like, could we use that same, like, endoscopy technology to look down the tubes of, I don't know, tube anemones mm-hmm. without, without trying to sample them and just, study them in situ?
Yeah.
Stuff like that.
Totally, yeah.